Oh je. Okay, can you hear me? Fine, good. So, welcome everyone today to our English lecture, kind of, in the great week of Wissenschaft in Kölner Häusern. Sie haben zu jedem Zeitpunkt die Möglichkeit, falls Sie etwas nicht verstehen, können Sie gerne meine Kollegin Frau Berghoff und mich ansprechen. Wir übersetzen auch gerne, falls es irgendwie Wörter oder auch Phrasen gibt, die jetzt nicht verstanden werden. Aber ansonsten werden wir diese 2 Vorträge auf Englisch halten und ich werde auch eine englischsprachige Einführung jetzt in die Alternsforschung mit Ihnen machen. So, I will switch back to English. And there the question is actually, why are we doing aging research? So, and there my colleague and I told before, Dr. Maren Berghoff and I, we are responsible for the communication of the aging research institutes here in Cologne. We have in Cologne a really big and excellent, actually, aging research community. So we have the CCAT Excellence Cluster of Aging Research, which has a lot of strong partners, also included there the Max Planck Institute for Biology of Aging, where my colleagues is from, and from those 2 institutes you will hear today a talk. But actually the whole cluster comprises also, of course, the university, since it's a university strategy of the Bund and Länder, the excellence strategy, and we also have the corporations there included, the uniclinics and also the Max Planck Institute for Metabolism Research, and also the German Center for Neurodegenerative Diseases. So why are we doing all that? Why are so many institutes interested in aging research and try to find there some treatments? That's why we have at the moment data graphs from North Rhine-Westphalia, and at the moment we have already the population of North Rhine-Westphalia has about 1/3 of the people living in North Rhine-Westphalia are over 65 years old. And in the upcoming years, this will rise to about there it is, to 50% of the people already in 2035, which will be over 65. So it is really important that we do aging research here in Cologne. And that might be not surprising to you. People are also getting older in North Rhine-Westphalia, and that's because, in general, all people are getting older, and the life expectancy increased over the last 100 of years. Okay, then I can move a little bit more. Okay, so living longer is really nice, but it also comes with a high price in some cases. So we have a lot of diseases which come with age, and with the risk for those diseases also increases with age. So here are just some examples. And the really important thing is that at the end, you might also not only have one of those diseases, so maybe you have dementia, but you also have on top of it also cancer. And the incidence for having more than one of those diseases also increases really exponentially when you get older. So when you are about 50, the incidence of having 1, 2, or also 3 diseases rise really tremendously. So, and that is also then a problem for our society, especially for our healthcare system. So, because when the people are getting older and older, that is also accompanied with more costs, because the people need more medication due to all the diseases they accumulated. So, what are we actually now doing in aging research here in Cologne is we have a different view on aging and also the diseases which come to aging, with aging. So before we have like the disease cancer, then cancer research is done, and then at the end you get, in the best cases, you have a treatment for cancer. And all those different disciplines were looked at it as similar. But we at the Aging Campus here in Cologne, we try to have a look at all those diseases with a common nenner. I don't have the English words right now. But we look at all those diseases due to aging, because aging is a really risk factor for all those diseases. And then we try to understand the aging process, to adapt that then to those different diseases which accumulate with aging. And then at the end, we try to find then treatment strategies which delay the aging process, or which make the, aging then the life we have later, the quality of life in the years from 50 on more qualitative. So, and with this short introduction, I want to introduce the topic of today, which is, as you read in the leaflets already, order is half of the battle. And that title is because a lot of processes are going on in our cells. And the processes are all highly organized, but with aging, they kind of are not that organized anymore, or certain other factors come into play and are disturbing the cells and doing their normal function when we age. So today we have two speakers from the Aging Campus here. We have first Professor Dr. David Wilches, who will tell us something about proteins and the function of proteins and what their role is during the aging process, and how they change also during aging. And then we also have a second, we have Chahat Mehra. I pronounced it wrong. No, Mehra, right? Okay, sorry. We have Chahat Mehra, who will tell you something about infections and aging, because this is also an important thing, because infections are, I know, I think we know all that right now, that because of the coronavirus, big infections can also get more dangerous when we get older. So, and with that said, I would give the words to Professor Wilches, and I hope, that works.
Thank you,
now.
Can you hear me? Yeah.
Yeah.
I hope I didn't.
Just a short note. So if you have questions, you can note them down on the paper we distributed. And but you can also, at the end, raise your hand, and then you can ask your questions directly. Another note on the COVID restrictions: you're allowed to take your mask off at your place. So if you feel comfortable, you can also take off the mask, right? Okay. So, and now,
Thank you, Anna.
Pointer. Pleasure.
Okay. So good afternoon. How are you guys today? Good. So, first I just want to say that I'm very happy to be here and have this chance to share our resource with you. So today I'm going to talk about our approach or our research to study aging and hopefully prevent diseases. So the goal of our lab is to understand how we age. So with that we can identify mechanisms that can improve the quality of life when we age, And with that, we hope to prevent or delay conditions such as disability or age-related diseases, like, for instance, Alzheimer, etc. So basically, we think that since aging is a major risk factor for many different diseases, if we target aging or we understand aging, that could be more efficient than just focusing on different diseases one by separate. So this is actually, this works? No. Okay, so this is Fauja preparing for a marathon when he was 100 years old, and that's exactly what we want to achieve. We want that people are healthy when they become old. So maybe this, I'm not sure. Doesn't work. No, I can use that, no problem. So, yes, so I have good news for you. So because the aging process can be regulated. So for instance, many studies in humans have demonstrated that aging can be influenced by many different factors. So as you can imagine, genetics is very important, and that has been also supported by studies in centenarians, so people that live older than, or more than 100 years. And of course, lifestyle is also very important. Conditions like smoking, exercise, or diet can also influence longevity. So if you ask me which is the most important factor, I would say that all of them are important. So I wouldn't have an honest answer here. So I'm sure that you know people that, for instance, smoke a lot, they don't do any exercise, and they eat a lot of McDonald's, and they still seem fine or healthy. So that means that genetics is, of course, very important, but I wouldn't take the risk of having a very unhealthy life. I wouldn't buy more tickets for the lottery. So then there are other factors that are also very important, like, for instance, it's known that females tend to live longer compared to males. And of course, social economic conditions or having a strong health system can be also very important for the longevity of the population. So, yes, so to understand aging and to identify mechanisms that can improve the quality of life, we need to do experiments. Of course, we don't want to do that in humans for ethical reasons. And of course, we also have to think that humans, the average lifespan for humans is around 70, 80 years. So if you are doing a thesis or a project on human lifespan, I wish you good luck with that. So, but I have also other good news, because many of the characteristics of the aging process are actually conserved, from invertebrates, from leader worms to humans. So for instance, conditions like altered communication between tissues, epigenetic alterations, genomic alterations, etc. Also a decline in stem cell function, and for instance, later I will talk about that, the decline in the quality of proteins is also very important for aging. And all of these characteristics are common in animals. So that means that we can use model organisms to study aging. So here we have a, yeah, maybe it will work. So here we have different models that we usually use to study aging. So as I said, human, they have a very long lifespan. But since we don't do experiments in humans, we can obtain cells from humans. And that's very interesting, because we can even do or transform these cells from the skin into stem cells that can differentiate into any cell type. So then basically you need to do research on neurons, then you can obtain neurons from these cells, or you need to study skin or muscle, you can differentiate these cells into the stem cells into the cell types. So then we have also my favorite one, which is C. elegans, this guy who is moving here around. We also have the fruit fly, Drosophila melanogaster. It works now? Yeah? Ah, perfect. Oh, no. Okay, I don't want to show the surprise. So, of course, mouse models are also very important in research. And we also have other invertebrate models that has been recently established, like the killifish. So now, if you guys want, we can play a game. So we can try to guess how these guys live. So Anyone wants to have an idea, for instance, how this leader worm lives? Don't be shy, you can say whatever. We won't judge. No? Very good. Very good. So, yes, so then, of course, we have the fruit fly that lives a little bit longer, and the mouse that lives around. Any clue about the mouse? Yeah. But we wouldn't do anything bad to them. I mean, we take care of them in the laboratory and not bad. Yeah. Quite good. Yes. So basically, C. elegans can live around 19 days. The fruit fly between 2 and 3 months. Mouse 3-4 years, but I think there are also some species. I mean, some mouse that can live 2.5 years. So it depends also on the species. And of course, killifish that lives around 4 and 6 months. And humans, as I said, 80 years. And cells that you can keep them forever in culture. So as you can imagine, one of the reasons why I'm so fascinated about C. elegans is the very short lifespan. So let me tell you a little bit about the story of the history about C. elegans as a model organism. So C. elegans was established, or was first used in a laboratory in the very far away now, 1963, and it was used by Sidney Brenner to study development by genetic screen. So he basically generated a battery of mutants. So then he could associate genes with defects in development, which is very easy to study in C. elegans. So after that, many important findings have been done in C. elegans. And just to give you an idea, 6 Nobel Prizes has been awarded to laboratories working on C. elegans. So, as I said, why C. elegans is a prominent or such an important model to study aging? So, first of all, they have a very short lifespan of 19 days, sometimes 2 weeks, as you said, depending on the conditions. But that means that we can know if our hypothesis is working or not in 2 weeks, basically, because after 2 weeks, you can already see if the worms are okay, or they are starting to die, or if they will live long. So that gives you a lot of time already to think if you need to change your hypothesis, or you can continue your project, so it's very convenient. So another thing is that many of the genes, human genes, are actually conserved in C. elegans. They are also expressed in C. elegans. So that means that we can do genetic screens in C. elegans to see if they affect longevity, and then after that we can test if they are also important for longevity in other organisms, like the mice. So another advantage is that C. elegans is a transparent animal. So that means that we can, just by using a normal microscope, we can see organs, even individual cells. So for instance, this is like how a plate of C. elegans looks like. So you will see that in the middle there is a drop, and this is basically the bacteria, which is the food of the worm. So it's also very cheap to do research in C. elegans. And then, as you can see in the normal microscope, we can see the different stages of C. elegans, like from embryo, larvae, adults, and even old animals. So then if we look closer, we can see individual organs and even individual cells. So another advantage, because since they are transparent, is that we can put a flag in each protein or in the different organelles, which is fluorescence. So then you can really track this protein during the aging process, how it changes, if it becomes mislocalized, etc. So, yes, so as an example, C. elegans has been very important to do groundbreaking findings in longevity. So one of the most important, or the most important maybe, it was in 1993, where Cynthia Kenyon discovered a worm, a mutant worm that lives twice as compared to the wild type. So basically she discovered that reducing the insulin pathway can extend the lifespan. And then other labs later discovered that this is conserved in other invertebrates, like flies, and even in mammals. And there is also some associations with humans. So these discoveries were very important because before that, everybody thought that aging is something that just happened. It's a random thing that happens with time because we accumulate bad things like bad molecules or proteins, etc. But this experiment show that aging can be regulated. So now is where scientists start to, to look for mechanisms that can make us live longer and healthier. So yes, so about our research, so we are interested on understanding how proteins regulate the aging process. So basically, our project is based on the basic observation that the quality of proteins declines or goes down during the aging process. So this decline in proteostasis can promote the accumulation of damaged, misfolded, aggregated proteins that affect cell function, and then eventually they can lead to disorders such as Parkinson, Alzheimer's, or Huntington disease. So what is a protein? I'm sure that many of you already know, but just in case. So proteins are macromolecules that are formed by amino acids that perform cellular activity. So any cellular activity that you can imagine, mostly they will be, or most likely will be performed by a protein or combination of proteins. And that includes, for instance, metabolic reactions, the transport of other molecules. Also, it's very important to maintain the structure of the cell, and also, for instance, to respond to stimuli. So we have to think that the protein network is super complex because each cell contains thousands of different proteins, and the protein content depends on the specific cell type. So, for instance, it's not the same the proteins that a cell from the skin has compared to a neuron or to a muscle cell, and also it depends on the status of the cell. If the cell is proliferating, dividing, or differentiating, so the protein content will change. So that means that proteins must be super regulated, tightly regulated, and adjusted for each cell type. So, but we know that many conditions, like stress conditions, normal metabolism, aging, or disease-related mutations, constantly challenge the quality of proteins. So these stress conditions can lead to the accumulation of misfolded proteins. So basically, proteins lose their normal structure, and then they become misfolded, and they are like a glue, or like a glue that bring other proteins. So they stick other proteins, and then finally they form aggregates. So of course, having aggregates is not good for the cells. And also can happen that some proteins become unnecessary because the cells they just need to do another function. So what to do with these bad proteins? So exactly what you do at home with your rubbish. So the cells try to eliminate them. So that means that the cells need to have a garbage system or a garbage truck to degrade these unnecessary damaged proteins. So for that, they use something that is called the ubiquitin proteasome system, where basically there's a guy here, an enzyme, a protein, that adds a flag to another protein. So then this protein can be recognized and degraded by the proteasome. There is a machinery that degrades or that trashes the protein. So here is a more scientific example or scheme. So here, basically what happens when a protein is misfolded or not needed, then the cell adds this ubiquitin flag to the protein. So then the protein can be recognized and degraded by the proteasome. But here there's another player that I would like to introduce, because sometimes after adding the ubiquitin and before it's degraded, the protein, then the cell can decide, okay, maybe I still need this protein, or this protein is important, I made a mistake here. So then they have the chance to remove this ubiquitin. So maybe, I don't know if that was complicated or no, but I would like to put this example about a wardrobe or a closet. So I'm sure that all of you have in your closet some clothes that are very off-season and very old, that you haven't used or wear for many years. And some of them can be even broken, or they can have stains of wine that you cannot remove, but you still keep them for some reason. So imagine now your mom comes or your partner and say, okay, I think we should make some room here. So then what your mom does is to put some stickers to mark these proteins, that the ones that you should trash. So normally what happens here is that you will go with the old clothes and then trash them or recycle them. But it can also happen that you say, okay, actually I don't want to trash this. This is my favorite jeans, and I'm going to remove the sticker and put it back on the closet. So that's what the deubiquitinase does. So since ubiquitination is such an important regulatory mechanism for cell function, we ask how ubiquitination regulates the aging process. So what we discover is that in the young animal, in young cells, the proteins that, the enzymes that put ubiquitin and the ones that remove are more or less balanced, so that eventually most of the proteins have ubiquitination flags, so they are marked for degradation, so they are properly ubiquitinated, and then they can be degraded. But when the organism becomes old or with age, then this guy here becomes more active. So then what happens is that it removes the ubiquitin, so then this protein loses the ubiquitination flag, and then it cannot be degraded. So basically what happens there is that the cell starts to accumulate proteins that they don't need or that can be toxic. So in summary, we found that with aging, there's a loss of ubiquitination, so then proteins cannot be eliminated. So then eventually many proteins accumulate with age, like IP2 or APS8. You don't need to learn or memorize these names, it's just an example. So then we ask if these proteins are important to regulate aging. So when they accumulate, they can be bad for the cell. And we found that actually we decrease these proteins by tricking the worms by genetic methods, then the worms live much longer. So for instance, if we decrease the levels or decrease the accumulation of IP2 or APS8, you can see this red line that they live much longer, even 5 days longer. So 5 days among the 19 days is quite a big percentage. So what is IP2? IP2 is a component of the intestine, of the fiber network of the intestine, and basically it's like the skeleton of the intestinal cell. So as such, is necessary or required for maintaining the integrity of the intestine. So what we found is that at day 1, IP2 is located where it's supposed to be, in the apical part of the cytoplasma. So they form this very nice structure. But then with age, IP2 levels start to increase because they cannot be degraded. And also, then it becomes mislocalized through the whole intestine. So you can see that it goes from this part to the rest of the cells. And on top of that, we found that IP2 forms aggregates. So as you can see, these intestines don't look so nice compared to the young animals. And we found that that was due to the aggregation and increased levels of IP2. So we know that a loss of intestinal integrity can induce the colonization of the tissue by bacteria. And for instance, this is very easy to track for us because we have E. coli that express a bacteria that express a fluorescence reporter, this red mark. So basically, we put this fluorescent bacteria on the plate, and the worms eat this bacteria. So as you can see, in young animals, there is not accumulation of bacteria in the intestine because the integrity is good, it's well established. But with age, maybe you cannot see very well here, but with age, there starts to be a colonization of the intestine, which becomes already very prominent in all animals. But if, again, we trick the worms to decrease the levels of IP2, we could prevent this bacterial colonization, which of course, the intestine starts to be colonized, but bacteria is not good for the animal. So by removing this IP2, we could prevent this pathological phenotype. So the other protein that I want to talk about today is APS8. So in contrast to IP2, APS8 is expressed only in muscle cells. And APS8 is very important to regulate the structure of actin filaments. which is again basically the skeleton of muscle cells. So as you can see, in young animals at day 1, actin filaments form these very nice fibers, but then with age, we start seeing alterations in the structure of actin. So as you can see, they become very disorganized and they form these weird waves. But if we decrease the levels of EPS8, we decrease the accumulation of this protein, we could rescue or prevent this collapse of actin filaments. So they look like as they were young animals again. So actin filaments are very important or they are associated to myosin filaments, which are essential filaments for the contraction of the muscle. So that's what let us allow us to move. So as you can see, similar to what we see with actin, the structure of myosin filaments also become disorganized with age. Actually, there's a breakdown of these filaments. But then, if we decrease the levels of PS8, we can bring back to the normal structure that we see in young animals. So finally, we know that. Okay. So finally, we know that alterations in actin filaments can also decrease the motility of the animal. So we found that indeed in day 1 animals, these guys move very fast, they are very happy. As you can see, they are very high activity. At day 15, when they are old, they barely move. But if we decrease the levels of EPS8, then they start moving again. So we can rescue this loss of motility. So to conclude, we have seen that in normal or in young animals, proteins are properly ubiquitinated. They have this flag for degradation, so then they are degraded by the proteasome. And then with age, these proteins become less ubiquitinated, and then they cannot be degraded by the proteasome. So then they start to accumulate across tissues, and they cause chaos in the cells. Like for instance, they induce many age-related phenotypes, like loss of intestinal integrity and loss of muscle function that eventually reduces the motility of the worms. So now, in our future plans, what we are trying to understand is the accumulation of these proteins, these regulatory proteins, can have a role in the development of age-related diseases. And with that, I would like to thank my nice laboratory, that as you can see is very international. We have almost from every single continent. And yes, also I would like to thank you for your attention. So with that, I conclude my talk. I don't know if we have questions now or later. Or at the end. I don't know. Yeah. Thank you. Oh.
Are there already some questions you want to ask now? Yeah? Mit der Angel, ne? Mhm.
For a micro.
I think I don't really understand the process of, I don't even know how to pronounce it. Right? I need glasses.
No worries. No.
Proteasome. Is that the cleaning itself, or I don't really.
So the proteasome is basically, so the ubiquitin is the flag, the mark, and the proteasome is the machinery that degrades, that cuts the protein.
Okay.
Yeah, so it's basically, I don't know, like in the garbage truck, when you put this machine that breaks everything. So I don't know if it's clear now. Yeah. Thank you. Thanks to you.
Any other questions right now? Otherwise, we will also collect your I don't English today. The handouts we gave you at the end. But now we are moving on to our next speaker, and we will learn something more about aging and infections.
Thank you. Oh, I think so. Right? Yeah. Perfect. Working. So Hi everyone, I'm Chahat. I'm not going to be talking a lot about aging, but more about infections, and it's going to be clear in a bit why. As the title suggests, our lab is interested in studying infectious diseases and why infectious diseases in an aging institute. So, what I have here on the left is a graph where you see the incidence of tuberculosis, which is a disease caused by bacteria. And you can see here you have different age groups. So here it starts with 0 to 4 years of age, and here at the end it's greater than 75 years old. And you can see over time there's this increase, gradual, in the number of people that get infected. And if you were to look at mortality, which is this table here, on the left you have different infectious diseases. Here again you have 0 to 4 years of age, 15 to 35, and greater than 75. And you can see clearly the older you are, the more there is a chance that if you do contract these diseases, that they can be fatal. So what we study is trying to basically understand what are the general processes, what happens when you are infected with the pathogen, and how the body fights this function, or how the body fights the pathogen. But before we start talking about it, just what is a pathogen? So a pathogen is any organism that can cause diseases once it's entered the body. And there are different classes. The three most famous ones are bacteria. Like I mentioned, tuberculosis is one disease that's caused by a bacteria. You have viruses. Thanks to COVID, we've heard a lot about viruses in the last 2.5, 3 years. And you can also have parasites, which I think are the coolest. I'm not biased, but that's what we work with. And I'm going to talk about one such parasite, and that is Toxoplasma. So here on the left, you can see an image of Toxoplasma. It's a single-celled haploid parasite. This is under a microscope. They're very, very tiny, so you can only see them under a microscope. And they can infect humans, and they're only able to grow once they've entered a cell. And a little bit more about how we can get Toxoplasma or how we can get infected is basically it's a cat parasite, so any feline species is the definite host. They can shed it in their feces, that can then maybe be eaten up the mouse, or come in contact with the mice, and then the cat eats the mouse, so that's how the cycle continues there. But it can also come in contact with different poultry or contaminate soil where we're growing vegetables, contaminate water, or you can get it from your cat litter, perhaps. If your cat is contaminated, then it can come in contact with humans. It's not very fatal for us, but it could be fatal if you're pregnant and you transfer it to your child just because they don't have a developed immune system, and so they can't really fight it yet. Here is just a worldwide occurrence of Toxoplasma. So you have different percentages, and every color represents a percentage. So here in turquoise, you have less than 10%, so maybe in China. Or you have countries where you have greater than 60% of the population, such as Brazil, or certain countries in Asia that are infected with Toxo. And just the point of this slide is that it's a very successful parasite, and it can infect a very wide range of hosts. So just some fun. What percentage of older people in Germany do you think are infected with Toxoplasma? So how many people think 20%? You can just raise your hands. Okay. How many people think 50%? Okay. And how many people dare to claim that it's 80%? Okay. So everyone's kind of right, depending on the age group, because here you have a graph from 20 to 80 years of age. And on left is just percent that have come in contact with Toxoplasma over their lifetime. And you can see as you age, you clearly have a very higher chance, almost up to 70% of having come in contact or being infected. So this also serves as a great model to try to understand what's happening once it's inside the cell, and of course then to see what would happen when you age and what goes on. So I'm going to talk a bit more about how we conduct our research. So we obviously have the parasite. As I mentioned, these are single-celled, and we use cells. So what David already mentioned, these are immortalized cells that you can use in culture, and we refer to them as host cells. So basically, you have the pathogen and the host. And when toxo enters, when it wants to grow, it makes an identical child, so as to say. And then you can go 1 to 2 parasites, 2 to 4, 4 to 8, and so far and so on, until at some point when it's reached a very big size, and then the cell bursts, and it can really burst out of the cell. I have a video at the very end. If someone's interested, I can show it. And then it's free again, and it can enter another cell. So if we talk a little bit more about how toxoplasma grows, so here you have a single parasite again, the image that I've shown before, and here you have multiple parasites, so it really is growing within the cell. And I also have a video, I don't know with the light, if you can see. No. How do I play it? There. So, sorry, actually, before I started, I just want to introduce it. So here we have toxoplasma labeled with red. So like David mentioned, you're able to put some color in, so we're able to visualize what's going on inside of our cells. And there's a cell that's infected, and you'll see here there is an 8-pack, and over time it's going to divide into... No. What's a presentation without technical difficulties? And you can see it divides, and it forms almost like a beautiful flower, at least in my eyes. And then it keeps dividing until at some point it's really big and it can burst. So what I want us to kind of think about is, well, basically, what does a parasite need to grow, and how does it get it? And of course, the parasite has its own machinery, which means that they can make it themselves, and that's something that we call synthesis. So they're able to produce whatever nutrients or building blocks they need to grow and continue. Or they can take it from us, which is in a way stealing, because we also have all the machinery and we make everything. And it's kind of, think of it like as takeout. Even if you have ingredients at home, you can just order in, and it's so much easier. So they can take whatever they want from us. And one such thing are fatty acids. So what are they? We consume lots of fat every day. And some are good for us, some are not so good for us. But once fat is inside of our bodies, it's broken down into something called fatty acids. And then these fatty acids can serve as building blocks for different things within the parasite as it's growing. And here is a piece of data. So let's look at the top panel first. You have uninfected cells. So because our parasites are red, we're able to see where the parasites are localized. You can see here that body P is something that's able to label all the fatty acids inside of the cell. And so here you can see that the fatty acids are distributed nicely in uninfected cell. And if you look here, just one cell, it's very nice all over the place. But as soon as you have an infection with toxoplasma, you can see all the labeling is localized to where the parasite is. And this is also clear here, suggesting that really they're able to take up all the fatty acids from our cells. So then, given what I've shared so far, the question then is, well, how do our cells really fight this infection? Or how do they fight the growth? And so for that, I think let's look at what all do we have inside of a cell. And this is a very busy slide. You have the nucleus here, lots of different organelles, but the organelle that we focus on is the mitochondria. And maybe you've heard of the mitochondria in the context of it's the powerhouse of the cell. So what does that really mean? And mitochondria can also take the fatty acids that I mentioned, which are basically broken down from different kinds of fat that we consume. They can take it and give us something that's energy. So energy is something that you need when you're walking, that I need right now to give this presentation, that you need for basically all processes ever. And so given that I've told you that they can also take up this fatty acid, what my boss was interested in trying to see, well, what are mitochondria doing in the context of infections? And what you can see here, this is an image of an infected cell with mitochondria and toxoplasma. So the toxoplasma here are in red, and the mitochondria are in blue. And you can see that upon infection with toxoplasma, you have mitochondria really close by to the toxoplasma vacuole. And by vacuole, I just mean the sac that keeps toxoplasma inside of the cell. And you can see they're really surrounding it all around. And I also have a video, so this is how you can see it in live cell. You have toxoplasma here in red, the mitochondria are in green, and if you just observe the movement of mitochondria, you'll see towards the end there is this nice accumulation of mitochondria around the parasite. So right here, this is the vacuole, and you see there's mitochondria going very close to the vacuole. And so the question that my boss asked really was, well, why do mitochondria surround toxoplasma? And the idea is that, as I had mentioned, you have fatty acids within a cell. And these fatty acids are available to both the cell or the parasite. And because, as I'd mentioned, mitochondria are really circling, they're really surrounding the toxoplasma pathogen inside of the cell, what she found is that actually from the fatty acids that are available, mitochondria are able to take up more, and then this decreases the amount that's available for the parasite. And that in turn restricts the growth of the parasite, and this helps our body fight infection against toxoplasma. And this I have here in a schematic as well. So basically over here you have mitochondria and different cellular organelles. Here I have fatty acids, but also different building blocks. And you have toxoplasma, and they almost have a tug of war fighting for nutrients, where mitochondria is able to take up more, decrease what's available for the parasite, and this in turn is able to decrease the growth. So just as a summary so far, toxoplasma, they live inside of their cells. They're able to take up fatty acids from their host cells. Mitochondria are actually able to block the access of some of these fatty acids inside of the cell. So this is kind of how, this is work from Lena, and this is kind of how our lab came into being. And for the last 5 minutes or so, I think I'd like to talk about what I'm particularly doing in the lab. And what I am interested in is, given that we see that mitochondria surround toxoplasma, how do mitochondria find toxoplasma? And this is mitochondria in a car. I can tell you for sure it's not GPS. They're just not putting it in and finding their way to the parasite. And why this question really fascinates me, because this has been published years ago. So you have here one cell, here in gray, and you have in green here a bacteria, which is also inside of the cell, and in red toxoplasma, and in blue mitochondria. And you can see the mitochondria really preferentially attracted to this parasite. And given that I mentioned that they have this kind of defense, or they're able to actually fight off the pathogen, what I'm interested in, well, how did they get there? So in trying to understand that, I wanted to try to come up with a system that would be a way for the cell to tell me, or for the mitochondria to tell us that, hey, we've reached. And in order to do that, we tried to use something that has been mentioned a little bit already today, which is using GFP, and that stands for green fluorescent protein, or just trying to create a signal or a color that will let us know that it's here. So if you're confused, that's fine. We're gonna go into it. So what is GFP? Years ago, I think 1960s, some really great scientist was asked a super simple question, which was, why are jellyfish green? They live underwater, but they can glow. And what they found is that they express this protein, which is green fluorescent protein, that gives them this luminescent quality so that you're able to see where they are. and this has over time been harnessed, so don't worry, we don't have jellyfish in the lab, and we're not still extracting the protein from them. At this point, it has been used, and now it's used for basic research, and this really revolutionized the field, because at this point, you can visualize any kind of process that's happening inside of our cells. And what we have done, or what I'm working on, is basically you have the protein that's green, but if you split this protein into half, you will have two non-fluorescent parts. So when they're far apart, they're not green. But when they come close by, it's green again. And so we want to use this for a system, because what I want is a signal for when mitochondria have reached. And so the idea is if we put one half on toxoplasma and one half on mitochondria, when mitochondria are close enough to toxoplasma, we should have a green signal. So I'm a visual learner, so I have a lot of videos. Here you have toxoplasma in blue, I hope you can still see it, and mitochondria in red. And I would like you to please focus in this area right at the periphery, or right around the parasite, and slowly it should turn green. Okay. So here already you can see a little bit of green coming on. Oh, come on. Okay. You look right here. You can see slowly. And then it's able to turn green. And so this is kind of what I've been working on. So my basic question is, well, mitochondria can find toxoplasma, but how? There are some other students in the lab, and they're also here. There is Lee, he's sitting at the back. He's looking at what happens at later stages of infection. Tanya, who's also here, is interested in, well, I mentioned that there's fatty acid as one source of food for the parasite. What are other sources? And Kavan is interested in perhaps, well, when mitochondria are so close to the pathogen, maybe they're producing something that's almost like a detergent that helps to kill them. And so he's looking at different antiparasitic substances. This is our lab. We're at the MPI of Biology for Aging. That's my boss, Lena, and that's the rest of us. So I'm open to questions. Thank you.
So are there any direct questions right now? Ja. Ja, die einen. He's going with that.
Thank you for the nice talk. How are you going to analyze the mitochondria now, since they have met the toxoplasma?
So
are you doing omics on?
Yeah. So what I performed is to do a CRISPR screen, and to basically knock out every gene in the genome and see what candidates would maybe promote this interaction and what candidates inhibit. I'm at the process of processing these samples and then hopefully send them for sequencing and then get some candidates. Yeah.
Okay.
Okay, thank you.
I think we will collect the cards and see what other questions you still have. Are there any other questions to the two speakers right now you want to address? Otherwise, we will have a look at the cards and then read them out for you. Stay.
Okay, should I do it?
Yeah.
Okay, so I will give the question to maybe both of you, and you can decide who wants to answer. Okay. David, you want to go up?
Oh, yeah.
Yeah.
Sure.
So here,
I thought you were
somebody
coming
asks,
too.
if
No?
is intermittent fasting, aside of healthy food, the solution to keep cells fit and flexible? I guess maybe we have to explain what intermittent fasting is. I mean, I don't know. I mean, the person who asked definitely knows, but intermittent fasting means that you have like a long period where you don't eat. So, for example, you eat the latest like 6 in the evening, and then, I don't know, 10, 12 hours until next day or even longer. You just don't take up any food, and then it means that your body needs to like regenerate and take up own energy. And intermittent fasting means that you don't do it for several days, that you don't eat, but just for a longer time during the day. And the question is, does it help to keep also the waste out? Maybe, David, that's a
So we
question
keep,
for you.
sorry, the question
The
is?
waste out, so like the great proteins.
I mean, actually,
What does it do to like lifespan, longevity?
I mean, it has been, there are several studies that show the autophagy is increased. So autophagy does some functions like the proteasome, so it can degrade proteins, but also organelles. And basically, it's important to recycle the material of the cell. So it can eat, it's like a autophagy, how we say, auto-eating system inside the cell that, for instance, get a mitochondria, eat it, and then it releases the what it was there, so it can be used again. So, for instance, in these lines, it has been there's some relation between autophagy and protein degradation and intermittent fasting.
Yeah, I mean, one of the most popular, I think, ways in aging research that people think to like improve health and old age is definitely diet and also intermittent fasting, right?
Yeah, but here what I would like to say is that, I mean, of course, in my case, I prefer to be happy and eat what I like. So maybe I shouldn't say that, but I think here is also, as scientists, one of the things that we are doing is that we can enjoy still things that, of course, things that are so bad. I mean, drugs and this, you know what I mean, no? But good food and this, that we still enjoy. So I think our job is to find how these things can actually extend your lifespan at the molecular level, at the molecular level. So then we just can directly modify this without sacrificing so much on the diet.
Yeah.
But I understand that there are people doing that, and
I mean, there are actually people doing it, but I mean,
yeah, by themselves. I know people who are doing that, and they claim that they are very happy and this, but I'm not so sure, to be honest.
I mean, maybe this next question really quite fits that, because you said like you're trying to find like a treatment or maybe drugs. So somebody else asking, so metformin, I mean, you know, maybe is one drug that is supposed to slow down the aging process. And there are some other experimental drugs and genetic methods that could counteract the aging. And should the society not discuss that these medications can be used as an anti-aging drug? So maybe you can comment on anti-aging drugs in general, and then, I mean, the societal question, yeah.
Yes, so I mean, what we do in the lab is mostly basic research to understand what is happening. Of course, if we can find a drug, eventually, I mean, we just can administrate and have a better quality of life when we age. That's great. So, but we basically want to understand what is happening, because if you don't understand what is happening, it's very difficult to find drugs. So, of course, there are several candidates, like metformin, resveratrol, also senolytics. Yes, so there are people even taking that by themselves, but many of them have still not been demonstrated that in humans are doing something. And yes, so there are some, like, for instance, rapamycin. So it's one of the classic ones that actually also activates autophagy. And yes, so there are people taking that and thinking that they will live long, but that's still not clear. And so actually, yes, there are some experiments that show that in some model organisms, under specific conditions, this can extend longevity, but in some cases are very minimal extensions of lifespan, and not in all the species. And also depends on the laboratory, so it's still, I mean, there's a lot of work to do. But of course, this is one of the main questions that there are many biotech companies and actually even rich people trying to establish these rich companies, investing a lot on that and looking for the perfect drug to make us live very long. And some of them claim to the immortality, but I think that's still very far to be reached, and there's still a lot of research to be done required for that. Yeah.
Yeah, and then maybe one last question. I guess that's maybe one for Chahat, because here somebody asked how to start a career in aging research if you're an applied biology student. I'm not sure what applied biology is, to be honest, but is that like a Studiengang? Ah, okay, I see. But maybe, Chahat, you can just say like how you maybe how it was for you to like start your PhD in aging research, and maybe we can give some recommendation.
Right. So I think something also what he was talking about aging. So when you're studying something like an infectious disease, it's between one pathogen and you can try to kill the pathogen and maybe you can cure the disease. But aging is so cumulative and there are so many different processes that come together that are dysregulated. So I don't think any one drug will be able to miraculously cure it all, because there's a lot that's going wrong at the same time. And so I think something like that, or something like a disease like cancer, you can really try to understand what's happening at the basic level so that you know when it's wrong, you can recognize it. Because if you don't do basic translational research and you don't know what the process should look like, you don't know what is going on when it's dysregulated. And so I think what I really found interesting was that there were a lot of different facets, like he showed in the beginning, there are all these different hallmarks. And one such is mitochondrial dysfunction, and that's something that the Institute of Aging is really trying to understand, because that's been shown in lots of different diseases that happen as you age. And so that's kind of how I got interested in it.
Did this answer the question? I don't know. I don't remember who. Yeah.
Maybe I can also give a remark on that. Like, when I understood it correctly, the person who asked the question is studying in Rheinbach, I would say. Because I did that also. I studied also applied biology in Rheinbach. And yeah, now I'm a science communicator. But actually, if you want to go to the aging field, there are a lot of... yeah, we like at the MPI for biology of aging, and also at the CCAT.
Yeah, I
like,
got people from there. They contact me for doing some internship in the lab and this. So
yeah.
Yeah.
So there are always opportunities if you go on the websites of those two institutes that you can then get in contact directly with the PIs and then ask if you can do your bachelor's, master thesis or so. And then that's, I think, your start into an aging research career, I would say, because there we indeed have in Cologne all the good scientists, all the brilliant heads in aging research we have here in Cologne at the campus, and also great infrastructure also. So we have really, you saw a lot of videos and fluorescent pictures, so we also have great infrastructure which facilitates your research here.
Yeah, so my advice is if you contact someone, don't be frustrated if they don't answer, because sometimes, I mean, they receive many applications, so, but that doesn't mean that they are not interested. Or, I mean, sometimes the labs are full, but so what I mean is that keep trying, because it's nothing about you or that. I mean, yeah.
Sorry, I think I also misunderstood the question, and I was answering more personally why I was interested in the lab that I'm in, but you can talk to me after and I can tell you the program that I applied through. So.
Okay, were there more questions? No? All right, then I would say thank you very much to our two speakers today. It was really interesting insights into the aging research field, to really different things, which also shows how broad aging research is. Like one focusing on more proteins, the other one more on toxoplasma. So it's really a wide field, the aging research field. And also, since we also heard a lot of aging today, I would also would like to give the remark that there's just next door an exhibition of a photographer who took pictures from aging people. So if you would like to also stick around a little bit more and then see what is the effect of aging. We heard a lot of about aging today, but then also to see how aging looks like in humans. You can join us and go next door and have a look at the picture exhibition there. And at the end, I would also would really like to thank all of you for coming. I know today with the weather was really not nice, and of course I would like to thank also the organizers who did this. Also the Kölner Wissenschaftsrunde, which organized the series, which was like the whole week. We are now at the end of the week already on Thursday. And also, of course, our host here, the Volkshochschule, which is really great that they give us the stage to show here some of the data of our scientists, and that we can also get in touch with you all and also discuss the aging research topics. This is really also great for the researchers to get questions and get input, and that they see what people are interested in. So thank you all for coming, and then I hope a lot of people join us in the exhibition. Thank you very much.